he received a PhD from University of Heidelberg, where he worked with Werner Franke, and there he started with ATPases. Then he moved for postdoc to Harvard Medical School, where he was with Mark Kirschner working on APC. And from 1996, he became group leader at IMP in Vienna. So from that time, I guess, he started to work on cohesin and SMC proteins. 2013, Jan became scientific director of the institute, and he organized SMC meeting in 2015 and 2019. So that's the background of his work on SMCs. I would like to ask him to present his lecture on cohesin now. Thank you.
I also, like the other speakers, would like to start by thanking you and your team for organizing this really interesting conference and for inviting me to speak. I think one can see from the number of participants who are out there, I don't know who they are, but there are many, that there is really great interest in this symposium and in this topic. I suspect that's a reflection both of the pandemic really limiting us in our scientific discussions, but also the SMC field being at a phase where it's extremely exciting. Now, in my talk, I would like to discuss whether cohesin complexes, about which we heard quite a bit of already, might mediate DNA loop extrusion and sister chromatid cohesion through distinct or related mechanisms. We don't know the answer to this, as you will see, but have interesting observations, which I would like to share. They're all unpublished and part of work that is ongoing in a project that's led by Kota Nagasaka in my lab, who you see here, supported by several people in the lab. One of them is Gordana Woods, who you can also see in the background, how she's supporting Kota, and others who are not on the photo, but whose name I will mention as I go along, in particular Roman Stoksitz, Ian Davidson, and Wen Tang. Kota, as part of this project, is also collaborating with the laboratories of Kim Naismith, Daniel Gerlich, and I think indirectly even with Adele Marston, although I don't exactly know where that stands, Adele. But I won't show, for time reasons, any of the data from these collaborations. Now, for the purpose of this talk, you need to know a few things which I think have all been mentioned already today by the previous speakers. I'll recapitulate them very briefly anyway. So like many of you, we believe, we don't know, but we believe that loops and tuts in interface chromatin are formed by cohesin by loop extrusion, and that this process would continue either until cohesin encounters CTCF molecules bound to DNA, shown schematically by these little triangles, or until cohesin would be released from chromatin by WAPO, which would thereby obviously limit the lifetime of these chromatin loops, unless WAPO was inactivated, which we think can happen both under physiological conditions, for example, in pro-B cells of the immune system, where this is thought to enable VDJ recombination, but of course can also be induced experimentally by depleting WAPO through various means. And in either of these cases, that leads to the formation of longer chromatin loops, because now cohesin has a longer residence time on chromatin, and by poorly understood mechanisms or unknown mechanisms really, under these conditions is able to bypass CTCF sites, and thereby to create not only more long-lived loops, but also longer loops, and might, in some cases at least, be able to eventually run into each other. So multiple different extruding complexes would encounter each other, and that then might lead to the formation of axial structures inside chromosome territories and interface, which one can see microscopically, and which Antonio Tedeschi, a former student in my lab, at the time when he discovered these, are called vermicelli. Now, you know, of course, that cohesin has other functions than loop extrusion, the most prominent known one being sister chromatid cohesion, where cohesin is not connecting two different loci on the same DNA molecule in cis, but is connecting two different DNA molecules, the replicated sister DNAs in trans to mediate cohesion and proliferating cells. And that observation in itself, I think, raises the very interesting question of whether or not these two functions, loop extrusion and cohesion, might or might not be mechanistically related. Now, there's a number of observations out there that would be consistent with the possibility that these two functions are not identical in mechanistic terms. Some of these reasons are listed here. That's not a complete list. One of them is, for example, the observation made by Kiko Tachibana that in mouse oocytes, the form of cohesin that mediates cohesion depends on Reg8, but the form of cohesin that is required for formation of loops depends on cohesin SCC1. Another observation made in Daniel Gerli's lab is that the genomic distribution of cohesin complexes that mediate cohesion in trans or loops and cis is overlapping but not identical, with the cohesive complexes being enriched at TUT boundaries, whereas extruding complexes appear to be present throughout TUTs, as we also just heard from Jo. And third, of course, Kim's observation or evidence that to mediate cohesion has to entrap DNA inside its ring structure. But then unexpectedly, Ian Davids in my lab found that a version of cohesin in which the three ring-forming subunits can be covalently linked to each other or are partly linked already to each other, so they cannot open and close, can still perform loop extrusion. So that would imply that the way that cohesin interacts with DNA might be quite different when it mediates extrusion as opposed to when it mediates cohesion. Now, if these two functions were mechanistically different, then one might expect that one would be able to genetically separate them. And so that is what Kota was interested in, and has therefore looked for mutants that might represent such separation of functions alleles. And I would like to talk about one of these, which Kota generated in human cells, being inspired by prior work that Madhu Srinivasan had done in Kim's lab, where he had found that two different mutations of cohesin's hinge domain, so the domain where the two SMC subunits heterodimerize, would abrogate the ability of these cohesin complexes to topologically entrap DNA, and presumably for that reason, were unable to mediate cohesion. But then at the same time, nevertheless, in these cells, RDNA loops could still be observed. These can be seen microscopically in mitotic budding yeast cells, consistent with the possibility that perhaps these mutants were defective in cohesion, which they clearly were, but perhaps not in loop extrusion. Now, the caveat with this interpretation was that it's not clear whether these RDNA loops are formed by loop extrusion, and a technical limitation has been that so far it has not been possible to reconstitute loop extrusion with budding yeast cohesin. So it was difficult to test that function directly. Whereas, as has been mentioned before, using human cohesin complexes, it has become recently possible to reconstitute their loop extrusion activity in vitro, as was done by Ian Davidson in my lab and several other labs that have been mentioned. Now, to exploit that possibility, Kota designed a hinge mutant in the SMC1 subunit of human cohesin by mutating 3 evolutionary conserved lysine residues into aspartates, and ask whether they would also be losing cohesion, as has been seen for the yeast mutants, and to be able to ask, could they still perform loop extrusion, yes or no? Now, to be able to test this, Kota developed a cell system based on HeLa cells, in which he is able to deplete the endogenous SMC1 protein by using auxin-induced degradation. If you add auxin, this endogenous SMC1 disappears. I hope you can see my cursor. But at the same time, he could express ectopically upon doxycycline addition an inducible version of SMC1, either in its wild type form seen here on the left, or representing this 3D mutant that I just introduced. And this system works quite efficiently in that the cells in which the endogenous SMC1 is degraded lose sister-chromatid cohesion, shown here, as measured by mitotic chromosome spreads. An example is shown here on the right. But that cohesion defect can be quite efficiently restored, or prevented rather, if one induces using doxycycline expression of a wild-type version of SMC1, which will then rescue that mutant phenotype. But if a 3D version of SMC1 is expressed, then you can see here that this cohesion defect cannot be restored. So clearly this mutant, as was true for the budding yeast versions, is defective in mediating sister chromatid cohesion. Now that's not simply because the mutant cannot bind to chromatin, because if one fractionates cells, one can see that the 3D mutant can associate with chromatin, although it doesn't do this quite as well or to the same levels as the wild type, which you see here, and that will become relevant later. Now, if one now looks at the viability of these cells by staining viable cells with crystal violet, you can see here that inducing degradation of the endogenous SMC1 by addition of auxin will kill almost all or virtually all cells. I don't know if there's any left. I can't see that here. But if the wild type version of SMC1 had been ectopically expressed before that, now cells will be able to survive. But in the presence of the 3D mutant, they wouldn't. So this mutant is unable to maintain or mediate cohesion, and presumably for that reason is unable to maintain proliferation and viability. Now, that in itself is interesting, and we're trying to understand why mutation of these 3 conserved lysine residues in the hinge prevents cohesion. But since this is a high C meeting and time is limited, I won't show any data for this and just summarize and then come to the genome architecture part of this presentation. What Kota has found is that this 3D mutant cannot be stabilized on chromatin in G2 phase, whereas cohesive complexes normally have a long residence time there to be able to maintain cohesion from DNA replication till mitosis. These complexes can also not bind sororin, which is a protein that is needed to maintain cohesion because it's helping cohesion to stay on chromatin and by protecting it from release by WAPO. But interestingly, even in the absence of WAPO, these 3D complexes are still not able to mediate cohesion, which implies that these mutants must be defective in a relatively early step in sister chromatid cohesion, possibly, we don't know that for sure, but possibly already in the establishment step. And so then one implication, of course, is that the hinge must be required for this process. That is, of course, not the first observation that supports this idea. Stefan, for example, when he was in Kim's lab, had very interesting observations which implied that opening and closing of the hinge might be required for cohesion establishment years back. Now, is this mutant able to perform DNA loop extrusion? That was the question why we generated these mutants in human cells. And that is something we can test in an in vitro assay that Ian Davidson in the lab established, in which he is taking lambda-phage genomic DNA, which is about 50 kb, or any other long DNA molecule that he can get hold of, and then physically attaches it at its 2 ends to a glass surface in a microscopic chamber, into which he then can, by buffer flow, introduce various proteins. For example, wild-type cohesin and NRPBL, which is required for loop extrusion. And by buffer flow, then the DNA will be stretched, and that makes it quite easy to visualize whether cohesin is able to form loops. And that is indeed the case for wild-type cohesin in the presence of NRPBL. You saw some stills already in Job's talk, where it would look like this, where the DNA is very rapidly converted into a loop-like structure. You can't see this in this particular movie where only DNA has been stained, but if one is fluorescently labeling cohesin and NRPBL, one finds that these proteins are sitting here at the base of the loop, consistent with the idea that they are extruding DNA, reeling DNA into these loops. Now, is the 3D mutant able to do that? The answer is indeed, sorry, next slide, yes. Here you're looking at a panel of stills from a movie of like I just showed you. In the top for the wild-type cohesin, where you see that the linear DNA is converted into a loop structure. And exactly the same is happening if we flow in the 3D mutant of cohesin. it can also convert these DNA molecules into loops. It does that with slightly reduced frequency. That's plotted here. This is the wild type on the left, reading on the right at comparable concentrations. And also at slightly reduced rates. It's a bit slower, but not very much, as quantified here. But clearly it can form loops. Now, if this activity, which is seen in vitro using purified components, is the same activity that forms loops in TUDs and cells, which we don't know, of course, but assume, then the prediction would be that the 3D mutant would still be able to support formation of loops in TUDs and cells. And to test this, we have performed Hi-C experiments and have found that that indeed is the case. So here, looking at the left at a Hi-C heat map of cells where the endogenous SMC1 has been replaced by ectopic SMC1. And then on the right, you see the same where the 3D mutant has been expressed to replace the wild type endogenous. And at first glance, these maps look pretty similar. But if you look carefully, you can see that the TUDs are a bit more faint. And if you look at these corner peaks, which represent loops, you can see, for example, in this upper left corner here, that more of these corner peaks can be detected in the presence of wild type cohesin than in the presence of the 3D mutant. And that's a reproducible feature, not just inter-experimental variation, and correlates, of course, well with these somewhat decreased levels of cohesin that we see in these cells. Now, if this mild, very mild Hi-C phenotype was caused by slightly reduced levels of the 3D mutant chromatin, then we might be able to restore that by depleting WAPO. And so that is what we have tried. For wild type cohesin, this has a rather dramatic effect reported by a number of labs before, where you now see many more corner peaks and more long-range interactions in TUD regions. A similar trend can also be seen for the 3D mutant, but the effect is much weaker. And in fact, if you compare carefully, and I'll show you quantifications for that, it turns out that the 3D mutant will generate Hi-C patterns after WAPO depletion that now look like the wild type cohesin would generate in the presence of WAPO. So WAPO depletion can indeed partially restore the mild hypomorphic Hi-C phenotype that we see with the 3D mutant. That's quantified on the next two slides, where we have counted the number of TUDs. They are hardly reduced or changed in the 3D mutant, as you see here, and also the insulation scores. So the separation of TUDs, as one can compute genome-wide, are virtually identical. That's different for corner peaks, as is shown here, where we have counted the number of corner peaks genome-wide. And you can see that for wild type cohesin, the number of corner peaks, you've seen an example just before, is dramatically increased. And if one looks now at aggregate analysis of these corner peaks sorted by the length of the loops that we think they represent, you can see that the length of the loops that can become detectable in the presence of wild type cohesin increases after WAPO depletion. Now, for the 3D mutant, that's different. We can count fewer loops, as you can see here on the left in the histogram. And in the absence of WAPO, that number is increased, but not to the levels we see in wild type after WAPO depletion, rather to the levels we see in wild type in the presence of WAPO. And that's reflected also in the aggregate analysis of the loops here. If you look carefully, you can see that after WAPO depletion, the signal intensities and the loop length distribution now for the 3D mutant mimic that of wild type. Now, that's interesting because it implies, on the one hand, that these mild phenotypes are caused by reduced levels of cohesin or chromatin, that can be restored by WAPO, consistent with the possibility that maybe the 3D mutant can be released more easily by WAPO, but of course there's other possibilities. But interestingly, it is in part reminiscent of phenotypes that have been observed in cells depleted of CTCF, where also the number of corner peaks is much reduced. That was first shown in 2017 by Alfei Schnorrer in Benoit Brno's lab and by Gordon Woods in my lab. I'm showing here in these panels data from a more recent paper that Gordon published last year, where she saw or confirmed that CTCF depletion here by ion interference reduces the number of corner peaks dramatically, more than we see with 3D, but the trend is similar. And here are example high-C maps where you see that some corner peaks become undetectable after CTCF RNAi and others become weaker. So in this respect, when it comes to the number of detectable corner peaks, the 3D mutant is, if you wish, a hypomorphic phenocopy of a condition in which CTCF has been inactivated. Now, to understand better what the properties of this 3D mutant are, we've done a ChIP-seq analysis and found that at most wild-type sites where we see, at most sites where we see wild-type cohesin, we can also detect the 3D mutant. But these are calibrated data. Mouse genomic DNA has been spiked in here for calibration purposes, but the signal intensities are always lower unless WAPL is depleted. Again, as in the essays before, that will now restore 3D levels to the wild-type levels. But what that was to be expected. What we had not expected and was therefore interesting was that after WAPL depletion, the 3D mutant, but not wild-type cohesin, also started to accumulate in different regions, which in many cases were regions where genes were convergently transcribed. You can see here one example here, where WAPL depletion leads to accumulation of the 3D mutant in this region of convergent transcription, whereas WAPL depletion does not have that effect on wild-type cohesin. And that's a genome-wide phenomenon. Roman Stoksits in the lab has taken all genes and aligned them here in a meta gene computationally, and then plotted the ChIP-seq reads onto this meta gene. And you can see that the wild-type cohesin has a peak at the transcription start site and then a little increase in the 3' region. Upon WAPL degradation, that pattern is enhanced, mostly at the TSS. The 3D mutant has much lower ChIP-seq read density down here, but is dramatically increased by WAPL degradation. But interestingly, now mostly in the 3' region, much more than the wild-type, even though it's starting from much lower levels, confirming that the two versions, wild-type cohesin and 3D, behave quite differently in response to WAPL depletion. This accumulation of 3D in 3' regions corresponds to new corner peaks that can be detected in high-C maps. Here's one example where you can see that after WAPL depletion, 3D accumulates in a region in which the wild-type does not accumulate. And you can see a high-C signal that's anchored by these cohesin complexes here, and presumably by a pre-existing site here. And that enrichment cannot be seen to the same degree in cells expressing wild-type cohesin, even though WAPL was also depleted. So some of these complexes that accumulate in these regions must be extruding complexes, we think. Now, what is interesting about this is that this phenotype is highly reminiscent of one that we had observed a number of years ago in mouse cells, in which we had depleted WAPL and CTCF, where we now found that cohesin complexes were accumulating at sites of convergent transcription, highly reminiscent of what Frank Ullmann had reported for budding yeast cells containing WAPL many, many years back. The big difference here is that for the 3D mutant to show this phenotype, all we have to do is to deplete WAPL, whereas for wild-type cohesin, both WAPL and CTCF have to deplete it. As if, again, the cohesin 3D mutant wasn't responding properly to CTCF, or was phenocopying, if you wish, a partial CTCF depletion condition. And so that means we have two different assays or two different phenomena where the cohesin 3D hinge mutant mildly phenocopies a CTCF depletion effect, namely in that it reduces the number of detectable corner peaks in high-C assays and accumulates in cohesin islands upon WAPL depletion alone, whereas wild-type cohesin would only do that if, in addition to WAPO, CTCF was depleted. And so the question is then, of course, what does this mean? Why is this hinge mutation not responding to CTCF like wild-type cohesin? One possibility follows from observations that Daniel Panne, Ben Rowland, but also Gordon in my lab made last year, which implied that CTCF helps to protect cohesin from release by WAPO at CTCF sites. And so we wondered whether the 3D mutant could be defective, could no longer be protected by CTCF from WAPO. And to test this, we have plotted all the ChIP-seq data that we have either onto non-CTCF sites or CTCF sites, and found that at non-CTCF sites, the amount of cohesin ChIP-seq signal, that's again calibrated, that we see upon WAPO depletion is indeed increased to wild-type levels after, to cohesin wild-type levels after CTCF depletion. But surprisingly, interestingly, that's not the case at CTCF sites. The 3D ChIP-seq signal is here. Upon WAPO depletion, it does go up. That's, sorry for that. It's very difficult to see. It's right under the wild-type curve here. Again, also in this assay, it goes to the levels we see for wild-type cohesin in the presence of WAPO. But in the absence of WAPO, wild-type goes even higher. So that implies to us that, yes, maybe the 3D mutant is partially defective in being protected by CTCF from release by WAPO, but maybe it's also defective in some other function or property that we do not know about in how it can interact with CTCF at CTCF sites. That's where we stand. So one key question we would like to answer, but it will take a long time to answer presumably, is what then is exactly the function of the hinge in cohesin's response to CTCF. But as a preliminary conclusion, that's why we started this, I think it's interesting to note that cohesin's ability to mediate cohesion is not essential for loop extrusion. And in that sense, the cohesion and loop extrusion function of cohesin can indeed be separated, at least into one direction. Whether a cohesin complex that is defective in extrusion would still be able to mediate cohesion, we don't know. We would love to know. We're trying to answer that, but we don't know yet. But into the other direction, clearly one can separate these two functions at this level of functional assays that we have. With this, I stop. Many thanks. I think I have mentioned everybody who, or the names were on the slides at least, who contributed, except for Gabi Litos, who has, my lab manager, supported this project in many different ways. I would like to thank all collaborators who are listed here and our generous funders. Would like to say that this was all done at the IMP in Vienna. We have a beautiful new building, and we are the founding member of the Vienna Biocenter, which is a great place to do science. And I'm happy to take questions. Thank you very much. I see a question from Jean-Paul, whether or not the 3D mutant is still acetylated. The answer is yes, but as judged by Western blotting, so we're looking at steady state levels, not to the same extent as the wild type, but yes, it is. There's a dramatic loss of sororin and a smaller reduction in cohesin acetylation. I'm reading a question from Judith.
Thank you.
Hi, Judith.
Thank you. Yeah.
Can the 3D mutant still bind NRPBR62? I don't think we've looked at binding directly, but given that it can do loop extrusion quite well, not quite as well as the wild type, but quite well, it must be doing that. Yes, we think. Otherwise, we know for sure that without an NPBL, which must be able to physically bind from all we know to cohesin, there would be no loop extrusion. So the answer must be yes.
From Ryotaro Kawasume, thanks for the great talk. Do you have any idea whether there is any connection between SMC1 3D phenotype and cohesin acetylation? For example, SMC1K648 was found to be acetylated in Chaudhary and so on. Can you answer this question, specific question?
I'm not sure I fully understand it. I can repeat what I said, namely that we know that the 3D mutant can be acetylated and is acetylated in cells, although not quite as well as the wild type. But then we do see a bit less of the 3D mutant on chromatin, and acetylation happens on chromatin. So it's hard to say whether that's a primary or secondary effect. I hope that answers the question. There's a question again from Jean Paul. Do you think that cohesion at CTCF sites may require some features related to cohesion establishment? It's a great question, particularly having heard Job's talk. I don't think we know the answer, but yeah, we're entertaining ideas like that. Yes, that's what I can say.
Hi, Jan. Great talk.
Hi.
I was just wondering, do you think that because there's no evidence that cohesion is at CTCF sites, that there's any link between CTCF and Sororine that compete with each other?
I don't think we have data on that. Yeah, I need to think about that. That's a good question. Yeah, I mean, there's of course evidence from Daniel Panne and Ben Roland that CTCF competes with WAPL. Yeah, we don't have data on that. I need to think about that. Yeah.
Great talk, Jan. Quick question. So it looks like the 3D mutant looks like a wild type without CTCF. Is there an effect on CTCF levels in the mutant?
Say it again. Is there an effect on
CTCF levels?
No. CTCF, from all we can tell, behaves normally. Yeah.
And now that you have a redistribution of cohesin, more at 3' ends, is there any effect on gene expression?
That we haven't looked at yet. Yeah. Given all the reports, including our own, on those effects, at least in HeLa cells being mild, I wouldn't expect too much, but we haven't done that experiment yet.